This is 6A, the key assessment, which is component 2. As maize seeds germinate, they produce the enzyme a-maize, which hydrolyzes starch in the endosperm to maltose. And you've done this experiment. You've seen this petri dish, and you've got we had seeds A and B, and this was stained with kind of a black colour. So in here we had agar plus starch, and then we flooded it with iodine, and it goes black. And then we had a seed here, and it actually had a clear zone around it. Whereas the other one didn't. They didn't work very well, but yeah, some of them did have a clear zone. And that was enzymes produced by the seed diffuse out into the agar and get to work on this starch. So the amylase produced, digest that starch to maltose, and then that's why it wasn't black anymore. So that was that, boiled and cooled. So this might be the boiled and cooled. So you've got that, and that's in your lab book. How you would develop and refine this practical procedure to determine whether the treatment of seeds with gibberellic acid, whatever that is, well, I think you know what that is, don't you? 'Cause you know a bit about the theory. Increased production of amylase. Right, well, how much detail do we go into? Do we give a full method? What do we say? Well, hopefully you're getting used to it now. You've seen this on a few occasions, particularly in your class question book, of, well, I'm going to give my independent variable, dependent variable, and some controlled variables as part of this story. So what is my independent variable? You don't get any marks for identifying independent variable. I'm going to write it anyway. So independent variable. Well, that is, some seeds are going to be soaked in gibberellic acid, and some are not. So some seeds soaked in 0.1 millimole per dm cubed gibberellic acid, and the other half will not be. What are we going to measure? So that's going to be our dependent variable. So we're going to measure this clear area, compare the size of the clear zone, measure the area, work out the area of this clear zone. So measure clear zone, and calculate area of clear zone. So for example, you could measure the diameter of this clear zone, for example, and you could make some assumptions, that the area is pi r squared, and then calculate that area of the clear zone. So that's what you're measuring. And then controlled variables, then we'd suggest a few of these. So same type of seed, like species, or for example, age, species, size, that sort of thing. And if we're soaking some seeds in gibberellic acid, well, the other seeds would be soaked in the same volume of of liquid that's just the water rather than the water with gibberellic acid made up. So same concentration. Let's talk about the start. So hang on, same time for soaking for water versus gibberellic acid. The same agar with the same starch concentration, so same depth, concentration of starch in the agar, and we don't only need 2 of these and we've hit our 4 marks, but I'm giving them all. Same, we always think about for any experiment usually affected by the temperature, so same temperature, and as these are plants you could have same light intensity. So 2 good controlled variables there. Right, we have a graph now, a rather scary looking graph. Just gonna have a little coffee whilst I look at this graph. Very nice, that's Dr. Schofield's new coffee machine. The following experiment was carried out to test the hypothesis that this gibberellic acid stimulates amylase synthesis, then causes germination. So you've got the acid, so the hypothesis is the acid leads to amylase, which leads to germination. That's what the hypothesis is. And here's a method. They were kept at 23 degrees C. That would help you with your method earlier if you couldn't think of anything. And they were counted at intervals after soaking. And then they homogenize them, that is, stick them in a blender and make them the same consistency throughout, and then somehow determine the amylase. So we've got a graph, germination. So germination is our little circles. That's germination. This sometimes helps in exams if you colour on graphs. And then we've got our amylase, which is this line here, which is our amylase there. Using the evidence from the graph, state whether you'd accept or reject the hypothesis. So, what happens first? Well, on this graph, we appear to have germination happening, and then only later, like a day later, we get the amylase increasing. So we've added the gibberellic acid, and then what happened? Did we get the amylase production and then the germination? No. We actually got germination happening first, then the amylase happening. So I don't think we should accept that hypothesis. So would we accept or reject the hypothesis? We reject the hypothesis. Or you might have said accept the null hypothesis, but it wanted you to say it like we said there. Why? Because those seeds germinated before there was an increase in amylase. So seeds germinated before the increase in amylase. And so that is AO3, which is where you're kind of evaluating the results of someone's experiment. But really, even though we don't know a lot about the theory maybe for this, it's just looking at a graph and interpreting it and relating it back to a hypothesis. So AO3, slightly more difficult.